Thank you very much, Katrina. So I'm going to give a talk which is not data-rich. It's historical, and I'd like to sort of basically talk a little bit about the history of the Asilomar meeting and what happened afterwards and its consequences for the field that we now call invasion genetics. So I'll start talking about the conference, and I'm sure some of you will know some of the facts of the matter. I'll talk a little bit about Baker and Stebbins because I knew both of them. I didn't know Stebbins particularly well, but I certainly knew Herbert Baker quite well. He was my PhD supervisor. And I went to work with Herbert Baker because I read The Genetics of Colonizing Species as an undergraduate and was completely turned on by its contents and thought this is the area I actually want to do research in. So I'll tell you a little bit about them. Then I'll try and define what I see, what is invasion genetics and what are the key questions. And then a little bit of self-indulgence, just because it seems very odd not to actually talk at least about some of one's research. I'll just give you some very brief vignettes on a couple of invasion problems I've worked on in my career. So, I think it's worthwhile, given that invasion genetics is part of a broader discipline that is now called invasion biology, to consider where did that come from? And there has been a little bit of controversy on that. I think most people in the field suggest that this was this book by Charles Elton, The Ecology of Invasions by Animals and Plants, was the sort of beginning of invasion biology. This has been challenged by Dan Simberloff. Not surprising, Simberloff likes to challenge things and he's very good at it. So he's not so sure. And in this book, which came out as the 50th anniversary of the Elton book, he writes a really interesting analysis of the history of invasion biology. He is of the opinion that Elton is not the founder of the field. He calls him the prophet. We can argue about terminology there. But he thinks it was the scope meetings that occurred during the 60s and 70s that at least got invasion ecology going. Now, as it turns out, if you look at those volumes, there's very little on genetics, so I don't think we can say that that was important for us. Interestingly, although this book says by animals and plants, there's virtually nothing on plants, and there's virtually nothing on genetics or evolution. There's a very brief mention of polyploidy and Spartina. He speculates on the cause of the decline of Canadian pondweed, Elodea, that it might have a genetic cause. And there is some discussion of the evolution of resistance in pests and fungi. But it's always seemed rather odd to me, given that Elton was at Oxford, and arguably Oxford is where ecological genetics was born, that Elton didn't take on board more evolutionary biology. And so, you know, if you read that book, it's pretty barren with regards to evolutionary and genetic ideas. So where did our field begin? Well, I think it could be argued that this volume that we're celebrating kicked this field off. But there's absolutely no question that at the end of the 60s and 70s, due to the electrophoresis revolution started by Lewontin and Hubby and Harris independently, working on Drosophila and on humans, the electrophoresis revolution allowed people to get out there and start measuring enzyme polymorphisms in plant populations. And pretty soon after that, there was almost an industry in going out, particularly in plants, because it was so easy to collect open-pollinated families of plants, to assay heterozygosity, allelic diversity, and so on. So it was the find 'em and grind 'em phase of the 70s and 80s, and there were many, many papers, and people like Bob Allard, Mike Clegg, Tony Brown, I think kicked off invasion genetics for plants at least. Then I think there was no question that the growth of conservation biology helped a lot with this sort of new enterprise. But from my reading, at least of the 70s, 80s, and 90s, as conservation biology and invasion ecology developed, there was a striking disassociation between invasion ecology and invasion genetics. And it's exactly the same disassociation that one can identify in conservation biology. And thanks to Russ Landy's seminal paper in Science, where he made an effort to point out that, in fact, these topics should be completely intertwined, but that there was a serious problem in that a lot of people were running around measuring heterozygosity and saying, oh, this is why the species is rare, Whereas in fact, often they were rare because of demographic factors. These were just symptoms of demographic problems. I think that paper was very important in making people think that clearly you need to have a complete integration of ecology and genetics if we're going to understand invasions. The new millennia saw lots of genomics and theoretical ideas and comparative biology, and we're going to listen to some of those today. And then I think the increasing urgency of the invasive species problem led to a whole new funding stream, particularly in North America, for people to start actually working on weeds and pests and diseases in basic departments. As somebody who did this at Berkeley in the 70s, I was a bit of a pariah going up to Davis regularly, you know, seen as sort of somebody, you mean you work on weeds? But now it's actually perfectly okay. We've just rebranded it, and now we call them invasive plants. Okay, so I think today invasion genetics is part of contemporary evolution, and it's part of invasion biology. Well, this is a photograph I took in 1973 in the field with Baker and Stebbins, and I'd like to tell you a little bit about these characters if you don't know anything about them. Herbert Baker was a very interesting person. He did his PhD in the UK on Silene, and he actually adopted the term invasion before Elton did. In his paper in Journal of Ecology in 1948, the term invasion is in the title, and he uses that word repeatedly as he discussed hybridization between a native Silene species and an introduced one. So he did his early work then on invasion biology, particularly invasion genetics, went off to Berkeley and spent the rest of his career there. He's probably best known for Baker's Law, but also he published many important papers in plant reproductive biology and on weed evolution. And in fact, I wanted to work with him on weed evolution. That's why I went there, because he was sort of, you know, the master in that field at the time. He also had a very close friendship with G. Ledyard Stebbins, who of course was much more well-known, the only botanist to be in the New Synthesis. Stebbins wrote this famous book in 1915, Variation Evolution in Flowering Plants, and was really the giant of plant evolutionary biology. They both loved to be together in the field, and I can say it was a real privilege for me as a graduate student to go into the field with them on a few occasions. They had very different personalities, and I think I can say this now. Stebbins was forceful, dogmatic, bit of a know-it-all. Baker, on the other hand, there was a lot of humility and was very much the junior partner who would quietly point out errors in Ledyard's reasoning at times. And Ledyard was actually very charitable to Herbert. Okay, so let's just briefly say the genetics of colonizing species. It occurred in February. We'd like to think it was actually in this building, but we don't know for sure. This isn't one of the older buildings at the Asilomar complex. It wasn't a big meeting. It was only 30 people. And given that there were 22 talks, that doesn't leave a lot of room for people who weren't speakers. So it was a pretty intimate group. I'm not going to read out the goal. You can read it there, but I don't think it's that different from the goal of this meeting. The volume contained 27 chapters, and authors were sprinkled right across the world, and there were no women, which was regrettable, but I think just a feature of the time, and clearly something that is not a problem at this meeting at all. Why was the book so attractive? Well, I think it was so attractive because of the heavyweights who attended that meeting, just like this meeting. That's supposed to you know, pick your favorite scientists. They're not all there, but there really was an absolutely star-spangled group of sort of luminaries in evolutionary biology with different perspectives on this problem. I want to tell a very short anecdote because this is something that's just happened a few weeks ago. I received an email from a Julie Mulligan, and if the name Mulligan doesn't mean anything, well then that's fine, but Mulligan was one of the authors in the genetics of colonizing species. He in fact was the only Canadian who was invited. He worked on the cytogenetics of Brassicaceae, and Julie Mulligan wrote to me and said her father was still alive, had seen the advertisement, he would love to come, but he was too infirmed, and that this meeting was the most important meeting of his career. It kick-started his career. He ultimately became director of the Biosystematics Research Institute at Ottawa. And in fact, if you're interested, Julie Mulligan has written a biography of her father, and there's a whole section on the genetics of colonizing species in this meeting, and you can get it through Amazon.com. It's called The Real Weed Man. Okay, so the thing that Herbert Baker was so pleased about when I was working with him in the 70s was the fact the book was still selling incredibly well and getting cited. And that has continued today. I mean, basically, that book is still features as a major citation in many, many papers in invasion biology. This is just a cumulative plot of citations through time. And I think it just reflects the fact that invasion biology has become an important area. So what is invasion genetics? I've sort of dug through the literature to try and see when was the first explicit definition, and it's by our friend Rob Coletti here, written as a graduate student in a paper in molecular ecology on the invasion history of spiny water fleas in the Great Lakes, and he defined it as the application of genetic techniques to investigate biological invasions. I've sort of expanded that a little bit in this definition. It's a mug's game, isn't it, definitions, But, this is my sort of slightly expanded definition. It's investigation of the historical, ecological, and demographic processes responsible for patterns of genetic diversity in populations and their influence on evolutionary change during biological invasion. And I'd be surprised if the vast majority of the talks today don't fit into that general sort of theme there. So this is a very bad slide. It's what you tell your graduate students not to do. It is entirely text-heavy, and like many of the speakers, I'm sure, who were worrying about time limits, I did have one of these things where I go through each one, you bring it up one at a time, and then it was like 7 or 8 slides, and I thought, my God, I'm just going to blast through this in one go. So I'm not going to actually go through this. These are just some of the questions that I think are now at the heart of invasion genetics. Pick your favorite question. You'll probably find one, and they range from the historical, such as the first question, trying to actually look at the historical introductions through using genetic markers, to patterns of population genetic structure and differentiation. A more general question in evolutionary genetics, which certainly applies to invasion genetics, and that is, what is the relative role of standing genetic variation versus new mutations to evolutionary change in invasive populations? Then in more the sort of realm of evolutionary ecology, how much local adaptation is occurring? Can we measure it? What constrains it? I know we'll hear all about these topics today and tomorrow. An interesting and hard problem that it'll be interesting to see whether anybody addresses is, does the evolution of local adaptation in invasive species actually increase invasiveness? That's been a much harder problem to sort of solve. We'll hear about the attempt to find whether particular reproductive life history and genetic traits tend to promote invasions. That goes right back to the heart of the genetics of colonizing species. There was much discussion in this book, as you know, in trying to come up with a kind of identikit picture, to use the sort of forensic metaphor, if you like, of a classic invader, just as we can try and do the same for psychopaths and rabid capitalists and other things like that. Are there certain traits that they have? And I think there's been a lot of work on that, and certainly Mark will discuss this shortly after me. And then getting again back to the genetics of colonizing species, what's the role of phenotypic plasticity during invasion? Is there such a thing as a general purpose genotype? Herbert Baker kind of coined this term, and we might ask today, well, do we have evidence for general purpose genotypes? What are they? I mean, is this a useful concept even? So, okay, so I'm going to finish just sort of with a bit of self-indulgence to look at some plant invasions that I've worked on over the last 30-odd years or so. And they're just like 4 little caveats. Rob's going to expand on one of them in his talk. But I'd just like to give you a sort of sense of why I was attracted to invasive species as sort of systems for studying evolution by natural selection, and try and sort of draw some lessons from each of these 4 sort of stories. And the first one actually developed when I was an agronomist working in Swaziland. Between doing my undergraduate and PhD, I got fascinated with rice fields and particularly mimicry, and discovered that there were these plants that were actually mimicking rice that were in the Echinoclea cruscali complex, and they had been the product of hand selection, essentially artificial selection by humans. Well, we could call it natural selection. In fact, I think it is an example of natural selection as opposed to artificial selection, where by mistaken identity, phenotypes of barnyard grass that looked dissimilar from the crop got pulled out. And over time, this moved phenotypes in rice populations to a full-blown mimic, which in fact is quite hard to distinguish from rice, particularly in the vegetative stage. The mimic is on the left, the cultivated rice is on the left, the mimic is in the middle, and then the congener from which it's evolved is on the right. And it turns out there are relatively simple genetic changes that allow this phenotype to look just like rice. A change in the anthocyanin pigment pathway, a block in that pathway so that plants don't produce red pigment. This, by the way, is a key issue for the hand weeders, because they look for the red coloration in pulling out barnyard grass, so any variants that are green. And then the simple genetic change, which has been used, by the way, by the breeders in rice, and that is to select in rice. The big advantage, the big breakthrough in rice breeding, as you probably know, and in many crops, has been the selection of upright leaves in cereals. And this, of course, means much better light interception down into the canopy. A lot of drooping leaves cause shade. And so, thanks to fantastic work by the Japanese particularly, they changed the whole crop canopy of rice, and the rice mimics have these upright leaves. And it would be very interesting to look at the genetics of that. I mean, there's been no genetic analysis of these traits. So I got fascinated with this group because Baker particularly had made a case that most weeds were generalist. I mean, this went along with this general purpose genotype idea. And I think we can say in the case of barnyard grass, yes, that's true. This race of barnyard grass is a real generalist. You find it everywhere. It's a real widespread species with broad ecological amplitude. But it has spawned these plants now that are restricted to rice fields. You never find them outside the rice field. They're harvested along with rice. They have no dormancy. And they're obligate rice field weeds. So it's a very specialized system. So that was one of the first things that I was interested in. I then got very interested in water hyacinth and particularly aquatic weeds. I mean, I was working in rice and, you know, looking in aquatic habitats a lot, and it was obvious that there were this whole group of invaders that got along just fine through phenotypic plasticity. Many of these populations have zero genetic variability. A classic example is Salvinia here, and thanks to fantastic work at CSIRO in Australia by Wendy Forno, she was able to harness the tools of biological control and actually has used this system as a fantastic example of how biological control can work when there's virtually no genetic variability. This is a sterile pentaploid fern. Okay, so there's actually no genetic variability in most populations. Water hyacinth was the species that I gravitated to because it had some interesting reproductive systems. And I'll just tell you that early work I did implied that there'd been a serious genetic bottleneck in its movement out of the Amazon, where it was native. And a recent paper in collaboration with two colleagues in China, where we sampled AFLP variations throughout the range, showed that, in fact, there was genetic evidence for this serious bottleneck. 70% of the individuals in the alien range, and these are sampled throughout Africa, North America, where it's introduced, in Southeast Asia, with the same genotype. So there's been a strong bottleneck, and I think phenotypic plasticity is the key here to water hyacinth success. It does extremely well because it has a very flexible phenotype. Now, because water hyacinth was a little hard to work on at Toronto, you need a lot of glasshouse space, you know, it grows quickly. I was looking for an annual relative that I could study breeding system evolution, and I'll just tell you briefly about the work we've done on Icornia paniculata, which is an annual species. This has been our main model system at Toronto for the last 20 or 30 years, and I'm particularly interested in it because I'm interested in the evolution of self-fertilization from cross-fertilization, and we've been able to show in this species there is evidence for multiple independent transitions from outcrossing to selfing. And what I want to talk about here is that that has been important in the evolution of invasiveness, because the area where this species is most predominant is in Brazil, where it's strictly outcrossing in most populations through a mating system polymorphism called heterostyle. But if you sample it in the Caribbean, it's highly selfing. So this is a sort of a classic example of Baker's law, where selfers are able to establish after long-distance dispersal. And what is particularly interesting in this species is that the change to selfing is a simple mating system modifier gene that alters the positions of stamens and causes autonomous self-pollination. The right-hand phenotype there, you can see the modified stamen, and that happens 24 hours before anthesis. The filament just grows longer, self-pollinates the flower, and you now have a selfing variant. And we've shown that it's different recessive alleles that cause this. So if we cross selfers from different parts of the range, we go back to wild type. Classical test of complementarity. So different recessive modifiers in different parts of the range produce the same selfing phenotype. And so from outcrossing in the center of the range, or the main, where most populations are, we go to the Caribbean. I want to mention here how modern work in sort of molecular population genetics can be used now to solve a puzzle. And the puzzle here was, are these plants really native to the Caribbean? This has been a long issue, and of course there had been the suggestion, since this plant was a serious invasive species of rice fields, it had come in as a rice seed contaminant. And I was a bit skeptical of this, partly because there's very little rice grown in the area where this species occurs in northern Brazil. And so one way of getting at this, of course, is to use coalescence simulations, and work we published, or Rob Ness, my graduate student, published in genetics, was able to show quite categorically that this thing has not been introduced to do with agriculture. Our Bayesian estimates suggest that, in fact, it's been there for at least 100,000 years. I mean, there are big confidence intervals on these estimates, but those of you who know the timing of agriculture, which is, 14,000 years ago, this obviously got to the Caribbean way before agriculture was even invented. And then lastly, the last story I just want to tell, and I'm not going to amplify this story because Rob will talk more about it, is in recent years we've been studying the invasion of purple loosestrife, particularly in eastern North America, where it's a very serious weed of wetlands. I wanted to show you photographs that I've taken in various parts of the world, and these are all native populations. And what's interesting in Europe, whether it be on the Tibetan Plateau, Portugal, the UK, Estonia, you never find large populations of purple loosestrife. But where we live, it's everywhere. And Rob has done some really outstanding work on this, which he'll tell you about, and I'll just motivate that work. The reason we were interested in it was because, for those of you who saw Chris Below's poster last night, it's still up there in the corner, we have very good evidence in this system of a very strong signal of stochastic processes through drift and founder events. And this plays out in the relative frequencies of the mating types that occur in populations. So we were clearly interested with this strong stochasticity that's going on as this thing increases in its distribution. Will there be evidence of local adaptation? And I'm not going to go through this slide in any great detail because it's essentially Rob's work, and I'm sure he'll cover it. But I think we have satisfactorily shown that these clines that occur in eastern North America are genetically based. They're associated with the evolution of local adaptation, and through reciprocal transplant studies and measures of selection, we've shown, I think to our satisfaction, that this plant has evolved as re-established clines in eastern North America over the last sort of 50 to 100 years. Okay, so to conclude, not all invaders are general purpose genotypes. In the case of the crop mimics, these are highly specialized weeds. Clonal invaders often rely on phenotypic plasticity, and often because of that, they can show very strong bottlenecks if they're introduced from one part of the world to the next. In the case of Icornia paniculata, the evolution of selfing can promote invasiveness through dispersal and establishment in novel environments. The ability to invade those rice fields in Cuba and Jamaica wouldn't have occurred if Icornia paniculata hadn't been able to get there through Baker's Law. And the re-establishment of clines in flowering time in introduced populations can evolve rapidly in response to climate. These are the people who did all the work. Thank you very much. I'd happy to take one question if there is one. No, there isn't one. Okay.